Hello everyone, today's Monday, so what you're going to have to do is the assign yourself, which is DNA discovery experiments with diagrams. I'll go over that after the do now, but for right now you have a do now, and you have a total of 7 questions, so I'll give you 4 minutes. You should be doing this by yourself, and if you get really lost, make sure you use your notes. So what I'm going to say for the sub, if you could go ahead and time them for 4 minutes. They should not be working with anyone else but themselves. I usually use my cell phone. If you want to use the clock, it's up to you. But definitely go ahead and time them for 4 minutes. And press pause on this video. Okay, time is up. So let's go ahead and go over numbers 1 through 7. So for number 1, it says that a cell with a normal diploid number of 32, how many would be in telophase 2 of meiosis? Well, since telophase 2 of meiosis is where we make sex cells, we're going to have half the number because half of you comes from your mom and half of you comes from your dad. So all you have to do is take 32, divide it by 2, and it'll be 16. Let's go ahead and look at our next 2 questions, 2 and 3. So we have different illustrations, and number 2 says which one has the least amount of DNA. So if I look at A, I have 4 sister chromatids. So 1, 2, 3, 4. B also has 4 sister chromatids. 1, 2, 3, 4. And if I look at C and D, the same thing. 1, 2, 3, 4, and D 1, 2, 3, 4. And the last one, E, only has 2 sister chromatids. So the one that actually has the least amount of DNA is going to be E. Then for number 3, it says, which diagram represents synapsis and crossing over of meiosis? And so the only one that we have the tetrads that are together and exchanging the information is going to end up being A. So A is where we have crossing over. This only occurs in meiosis. That's the only one where we want the mix of DNA. Let's go ahead and look at 4, 5, 6, and 7. So for number 4, the answer should have been prophase of meiosis. That's only when we have crossing over. And number 5 is, how does prophase from mitosis differ from prophase of meiosis? Well, crossing over is a characteristic of prophase 1 of meiosis, but not mitosis. Mitosis, we want everything to be identical because those are the cells that are going to help us grow. If we had cells that were all mixed up, we would look like Picasso. That would be crazy. So for meiosis, that's the only one where we're going to have the crossing over so that we can get the mixture of DNA. Number 6 says when a cell contains half the number of chromosomes, it's called a haploid. Why? Because hap sounds like half. For this one over here, number 7, this one was really tricky. So if you got this one, like, wow, that means you answered all the questions fast. And that means that you understood this trick question. So this says, how many copies of chromosome 13 did Ludwig van Beethoven inherit from his father? Well, you only have 1 set of DNA from your mom and 1 set of DNA from your dad. So he only got 1 chromosome from his dad. So now that we've went and done the do now, let's go over this. What the heck, Miss De Leon? Where are you? I'm in the upside down. Woo, woo. No, I'm just playing. I'm actually at a training. I have a training today and tomorrow. It's going to be at the Nat Medical Center. If you need something you don't understand, I have my cell phone that's going to be written on the board. Obviously, it's not on this video because this video is going to go on YouTube. But text me if you need something. My people that have their November 15th deadlines, I see you on my common apps. I'll make sure I get them done on time. I am going to say this, as much as I would love for us to just do easy work while I'm gone, we have a very, very mini unit right now, and we have to take the test on Monday. So we have to keep going today. There are things that we need to do. So today's agenda, we have the do now. We have a concept map that you'll get to work on, and this concept map, you can actually use it with your meiosis quiz. And the harder you work on the concept map, the more prepared you'll be for the quiz. So I hope that you guys get some 90s and 100s. I'm really excited about that. And then we also have DNA notes. If you have any extra time, you can go ahead and work on your assign yourself. All right, let's go ahead and keep going. Today's quiz is going to be an FRQ on meiosis. So on the quiz, you're going to have to describe all of meiosis. You can either draw it, but if you draw it, it has to be labeled and you have to add little tiny descriptions. Or you can just write it all out in paragraph form. And the great thing about today's quiz is that you get to use your concept map. So I know this doesn't usually happen. I don't usually let you use notes on your quiz, but I'm not there in class to guide you. So I at least want to meet you in the middle. So this is what we're going to do. This is going to be your review for your mitosis meiosis quiz, but really it's meiosis. You're going to have 10 minutes to work on this map. So what you want to do is you want to just branch off of it, and you can either describe it, or if you're going to go with the drawings, you can go ahead and draw, label, and a little quick description. When you work on this with your group, you can only work with your group, and you guys must be sitting down. The concept map must be turned in with the quiz. So if I don't get the concept map, it's 50 points off the quiz. So in other words, the only thing this concept map can do is actually help you. I predict that if you and your group work very hard in filling out all the information from interphase all the way to telophase 2 cytokinesis, that you will get the 100. I am going to tell you this, interphase, this set is going to be in the cell cycle notes. It's going to be in part 1, whereas like meiosis, prophase 1 to telophase 2 is going to be in meiosis part 1. So with that being said, you have a total of 10 minutes to go ahead and get this concept map done. So my sub, if you can go ahead and press pause and then time them for 10 minutes so that they can go ahead and work on the concept maps. As soon as the 10 minutes are done, we have to go into the quiz because I still have notes to go over with you. So with that being said, I will go ahead and let the sub put 10 minutes. Go ahead. Okay, let's go ahead and keep going. So now that time is up, we're going to go ahead and take the quiz. So the sub is going to go ahead and pass this out. On your quiz, you're only going to have 10 minutes to complete it. Remember, you can either draw, label with small descriptions, or you can describe this. You can describe this in paragraph form. It's really up to you, but you have 10 minutes, and you can also use your concept map. So if the sub can go ahead and pass out the papers, and then they should only be using their concept map. Notes and note cards cannot be used for this. You can go ahead and set your timer with the phone or the clock, but please give my students 10 minutes to go ahead and get this done. Thank you. Okay, time is up. So let's go ahead and pass our papers in. Please make sure that your name is on both things, the concept map and on the FRQ. Place your concept map on the FRQ and pass it to the right. If you try to pass one of those in at the way end of class, the sub should not take it. Everything should be passed in right now. So put the FRQ on top and then pass it to the right, please. I'll go ahead and let you guys do that. Pass it in, pass it to the right. Let's go, chop chop. Okay. All papers should be to the right. So I have a few things that we do need to go over. The assign yourself. The assign yourself is to go over like the experiments, but the experiments you get them from the notes that we're doing today. So on your notes, if you look at them, this is going to be like page 1, this is the back of page 1, and then this is page 3. On the other side of page 3, you have an area that says summarize. This is what your homework is. You're going to have to summarize by describing the experiments or the contributions of each of the experiments or scientists. And then I need like a little diagram, like how will you remember these experiments? So with that being said, I have one more thing, 2 more things to go over, announcements and celebrations. Your catalyst today is going to be with your homeroom for this week. We still have our exam on Monday, November 13th. That has not changed, which is why we still need to take notes today. Anyone that hasn't taken the Unit 3 exam, I know I told people they could take it with me on Monday, but I'm not here. So I'm very sorry about that. We can do this after school on Wednesday. So my people that haven't taken Unit 3 exam, the cellular respiration and photosynthesis, please meet with me after school on Wednesday so that we can go ahead and go over it. And then, yeah, that's what we'll go ahead and do. For Unit 4, I'll be tutoring that on Thursday. So tutoring for Unit 4 will be on Thursday. With that being said, let's go ahead and scoot on along. For celebrations, woo, 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 woo. Celebrations to Luis and Mike and Nelson and Jolisa for getting into Emery and Henry. Woo! Let's all clap, everyone. Or don't leave me hanging, people. Yay. Yay for them. And some even got money. Oh my goodness. I heard someone got like $20,000. That's amazing. If you got accepted to a college, please let me know. I want to celebrate you. I know I'm not going to be here tomorrow, but if you text me like so-and-so got into this college, I would love to like put you on the PowerPoint so that I can celebrate you. So I'm so excited for you guys. This is so exciting. I can't believe it. We're getting into colleges. So I'm very happy. Please let me know if you guys get into colleges. So happy for you guys. Okay, now that we're done with that, now we can go ahead and start the notes. So today we're going to talk about the structure and the function of DNA and all the scientists and all their experiments that led to this. So this is just a review. Which two biomolecules can you actually see in a chromosome? So let's see. If I look over here, I have DNA, that's a nucleic acid, and these are going to be wrapped around proteins, and proteins is a histone. So I have two. I have proteins that are the histone, and then I have nucleic acid, which is DNA. Let's go ahead and move on. So let's first talk about Morgan's experiment and what it revealed about genetic material. So Morgan had a fruit fly experiment, and it revealed that chromosomes carry genetic material. Since chromosomes are made of both DNA and protein, scientists knew that either DNA or protein must be the genetic material. So they figured it out. Okay, it's not going to be carbs. It's not fats. It's going to be either DNA or protein. So let's say we had to make a prediction on this experiment. This says, what would happen if 4 of these were injected into mice? So this is what we have. In these cells, these are living and controlled. These ones are going to be the ones that we didn't put anything in. These are living R. These are the harmless bacteria. So this one is pathogenic. And then this one right here is heat-killed, that they were bacteria that were killed, but they were pathogenic. And then you have a mixture of heat-killed, of pathogenic, and of harmless. So what do you think would happen with all of these? I don't know, Miss DeLeon. It's tough. Talk to me about it. Okay, let me tell you. So what ends up happening is if we took this really bad bacteria, it's obviously going to still be deadly because it's living. So this means that it's like living bad bacteria. Killed the mouse. Over here, we have harmless bacteria. So obviously, if you put harmless bacteria in a mouse, it's not going to do anything. We have tons of harmless bacteria in our digestive system, and they don't kill us. And then if we have this one, this says heat killed of bad bacteria. It's not going to kill the mouse. The mouse stays healthy. Why? Because the bacteria is dead. But let's say I put a bunch of dead bacteria and harmless bacteria. The mouse freaking died because the DNA of these bacteria were passed to the harmless ones. That's crazy. Okay, well, let's go ahead and see about Griffith's experiment. So now we're on to the second experiment. So Griffith injected mice with live bad bacteria, and they died. That's really sad. And then he injected mice with harmless bacteria, and of course they survived, right? I mean, that makes sense. It's not pathogenic. Then we took dead bacteria and we put it in a mouse, and the mouse survived because all of the dead bacteria was, well, it was dead. And then you have a mixture of the really bad bacteria with harmless bacteria. And what ended up happening is the mouse actually died, which was very surprising for them at the time. So, what's the significance? Griffith's experiments show that the biomolecule in dead bacteria cells carry genetic information, and the genetic information could transform harmless bacteria cells into pathogenic cells. So, for example, we can take bacteria and we can transform them, like we can make them glow. We can transform them to pass good genes on. We can transform them to work for us, like in antibiotics, but we can also transform them to be extremely deadly. And so we realized the thing that was causing transforming good bacteria to bad bacteria was because of the DNA. So let's look at this. I know we haven't talked about viruses since 9th grade, and we saw this in 7th and 9th grade. The review is, how do viruses or phages infect bacteria? So if I look, I have this little bacteria, and no, it's not pooping or peeing, although it looks kind of weird, right? Like to me, it looks like that. Gross. It's actually putting DNA inside of a cell. Oh, that is spooky. Ooh. Okay, let's go ahead and keep going. So what did scientists know about bacteriophages? So scientists knew that bacteriophages could actually infect bacteria with their own genetic material, causing them to produce more viruses. So for example, we knew that the reason why like someone gets AIDS, it's because of genetic material that was being passed. So the thing is people didn't know, is it proteins that's being passed or is it DNA? So this is an example of an experiment, but I want us to take a look at some of these questions. So this is what this says. Which macromolecule contains sulfur? DNA or protein? I'll give you a second to decide. Okay, the answer is protein. Only protein could possibly have sulfur. Miss, I forgot that. Okay, that's okay. We're still going over it. Which one contains phosphorus? So when we say like chon P, which is the one that has the phosphorus? Okay, well, that is going to be DNA. This says when you grow bacteriophages in a flask with radioactive S, where will some of the sulfur end up? And then we would expect that it would end up like in the cells. When you grow bacteriophages in a flask with radioactive phosphorus, where do you think some will also end up? Well, we think that it'll probably end up like in a bacterial cell. And how could you use this experiment to set up and test whether DNA or proteins are the genetic material? Well, if we know that phosphorus is in DNA, then we would expect only phosphorus to be the one that will show up. But if it's going to be sulfur that ends up showing up, then that means that it's protein. So in case I kind of lost you in this, I know that I was throwing a bunch of words around. What we're trying to see is what's going to pop up. Is it going to be sulfur, which is only found in proteins, or is it going to be phosphorus, which is only found in DNA? So that's what we're trying to figure out. Miss, this is confusing. I know. This entire thing today is AP Bio. So I'm going to skip this just so that I can like go over the description. So this is called the Hershey and Chase experiment. So Hershey and Chase infected bacteria with two types of bacteriophages. So you had phages that had sulfur, and these were incorporated into the proteins. And then you had phages with radioactive phosphorus, and these were incorporated into the DNA. So let's go ahead and keep going. So what does this mean? Well, bacteria infected by viruses contained radioactive phosphorus. So in other words, the phosphorus showed up, which means that it was DNA that was passing the information. This was huge. Like for us, it's simple. Like you and I are like, well, not you and I, but like all of seniors should know that DNA is the genetic material. But back then, we didn't know this. This was a very big thing for them to find out. So what was the significance? The results show that that viruses injected DNA, not protein. And so this meant that DNA is the genetic material, not proteins. This was like huge back then, guys, because we didn't even know this. Like such a simple thing for us to know. We had no idea. So Chargaff, I need to go over the discovery of Chargaff. Chargaff determined that the amount of adenosine to thymine, and the amount of cytosine was equal to guanine. So in other words, A's paired with T's, C's paired with G's. Ants go with tías, and cats go with gatos. And so this suggested that the nitrogenous bases paired up with each other. He also determined that there's a percentage of each nitrogenous base that varies between species. So for example, if for every A there's a T, that means in DNA, if you have 10% of the DNA that is adenosine, then that means that 10% will be thymine of the DNA. So if I took like a long strand, and this strand was like, I don't know, as long as the room, and I told you, okay, this strand, 30% of it is going to be cytosine. Then you say to yourself, okay, I know cytosine is going to pair with guanine. So if I have 30% of my strand as cytosine, then that means 30% of my strand should be guanine. So for example, let's look at this question. If a sample of DNA contains 10% adenosine, what are the percentages of the other bases? Well, then 10% would be thymine. So that means 20% of the DNA is adenine and thymine. And so therefore 80% of it is cytosine and guanine. And this is something that we'll keep going over. So if you're kind of saying like, it's a little bit tricky, this is definitely something that I want to keep going over because it might just be a multiple choice question that pops up on the AP Bio test, but just to be safe, just to be on the safe side. So let's talk about Rosalind Franklin, Watson, and Crick, what they discovered. So using X-ray crystallography, Franklin discovered that DNA is a double helix with a sugar phosphate backbone on the outside. And it has pairs on the inside and can contain infinite different sequences of the bases which allow it to carry the genetic information. This was huge. We had no idea this was occurring. I hope my face isn't in the way. Is my face in the way? Oh, geez. I hope it's not. I'll print out like an extra copy for the sub, just in case we're missing some words. Okay, let's go ahead and keep going. So this is what we have. This says label the following as complementary nitrogenous base pairing and the sugar phosphate backbone. I actually want to go ahead and make licorice with you guys on Tuesday. How we, the candy ones that we did in 7th and 9th grade that we got them and then we twisted them. If I hear that this went really good for the sub on Monday, then I'll go ahead and make everyone like little baggies of licorice and marshmallow and toothpicks, and I can have you make it while I'm kind of coaching you through it. So we'll get to the DNA. So let's come back to this. What did Rosalind Franklin, Watson, and Crick discover? So Watson and Crick used Franklin's data without her knowledge or permission to build a physical model of DNA, and they got tons of money. They got like $1 million and the Nobel Prize for the discovery. But get this, Franklin didn't get any credit. So check out these FYIs. Watson and Crick's first DNA model was 3-stranded and inside out. So actually they were wrong. And then Franklin's lab partner, who never liked her because they said she was too forceful as a woman, showed them the photographs. She died before the prize was awarded, so she wouldn't have been eligible, but still, it wasn't right of them for what they did. And you know what? She died from too much x-rays. That's crazy. She had cancer all over her body from head to toe because she disrupted so many of her cells that her DNA was all screwed up. So it's so sad. But let's not cry. Let's keep going. So these are all the scientists that we've mentioned this year. The ones that we will also mention. Some of them you might not be familiar with, but we will mention them as we go on. But for right now, what I want you to do is I want you to go ahead and summarize all of the experiments. So I'm going to go up on my little PowerPoint so I can go back to... Let's see. Let me get back to the thing so that you know what to do. So now that you have a little extra time, if you have the extra time, I want you to go ahead and work on the homework. And make sure that you get all of this done. And if you don't get it done in class, you need to make sure you get it done for homework. Use your notes, please. And if we did super awesome and the sub is like, yeah, they did great, then we'll go ahead and make candy DNAs, and we'll just go ahead and do this like back and forth. And on Wednesday and Thursday and Friday, we'll review for Monday's unit exam. Thank you very much, and have a beautiful day, guys. Please let me know if you've been accepted to a college. And goodbye.